remarkable adaptations out there to very hot places, to very cold places, to places at the bottom of the ocean, to the tropics. And in many places, that extreme environment is constant. And so another really remarkable thing that we see is that there are organisms out there that are evolving in environments that are not constant. And so today I want to really focus on the impact that a changing environment can have on alleles that are being experiencing evolution in those environments. And I really want to broaden our mind to thinking about environment as not just the abiotic environment or the biotic environment that organisms experience outside of themselves, but also that there's an important environment that an allele experiences that is its own genomic background. So genetic environment interactions, but also gene-gene interactions are an important context in which selection acts on alleles and populations. And of course, it's not just the abiotic environment or your genomic background, but also the type of an organism may change the relationship between an allele and fitness. So in my group, we work on a lot of different systems where we're trying to understand and characterize how genetic variation impacts fitness, but really what we work on are the physiologies and the phenotypes that we think link the genome variation to fitness. So we try to make that connection in the context of changing environments or different environments. And by making that connection, what we really hope is that we can generate hypotheses about how genomes evolve. Because of course there's this reciprocal path, and that's how phenotypes that impact fitness, how those shape the experience of the genome to natural selection. And so we work on this in a number of different systems. We work on some butterflies and the physiology of their overwintering processes with very different overwintering strategies like migration versus diapause. We work on the evolution of plasticity, so gene-environment interactions are really important for the evolution of phenotypes. We've worked on that in both Drosophila and now working on that in monarch butterflies. Of course, phenotypes differ across temperatures, particularly for ectotherms, and so we work on the evolution of thermal performance curves, and this is really a project that parallels quite nicely some of the lab evolution that's done at your institute, and we do that in flies, but also have started working on paramecium. And then today, I'm really going to focus on the genetic work we've done that links genome variation in two distinct genomes, the mitochondrial and the nuclear genome, to various phenotypes and fitness. And it's in this system where we've really been able to then generate some hypotheses about genome evolution that we can test. And then we're starting to expand this work out of Drosophila and into this little, in fact, very tiny mud snail from New Zealand. And I'll talk a little bit about that project, but that's a new direction for mitonuclear coevolution in the lab. So gene-environment interactions are really important for how an allele experiences selection. And that how it experiences selection can change depending on what environment it's expressed in and what its genomic background is. And so today we're really going to focus on the effects of the abiotic environment on mitonuclear variation. And then I'm going to show you the importance of what I'm going to call the phylogenetic environment, or the taxonomic group in which genomic mitonuclear coevolution is happening. And that will relate to modes of reproduction in this snail. And then in the last part, I'm going to show some really new data on genome evolution, showing that the mitochondrial genome itself may be a very interesting environment in which alleles are experiencing selection. Okay, so why focus on mitonuclear coevolution? Well, that really goes back to the physiology that's happening in the mitochondria. So inside the mitochondria is where the electron transport system sits. These are the complexes of oxidative phosphorylation. This is the process by which electrons are being moved through and ultimately accepted by oxygen. And the result of that is that protons get pumped out of the mitochondria. And of course, it's that proton gradient that then drives the formation of ATP. And this is why aerobic eukaryotes can get so much ATP production from breathing oxygen. But that entire process requires that there are proteins that are made from two distinct genomes in our cells, from the mitochondrial genome and the nuclear genome, and they come into quite close physical contact with each other. And so it's for this reason that people have been interested in the potential for genetic interactions between variation in mitochondrial encoded and nuclear encoded genes, whose protein products have to interact with each other. But in fact, it's not just interactions between OXPHOS. Essentially, all of the functions of the mitochondria are requiring interactions between gene products of the mitochondrial and nuclear genomes. So mitochondrial transcription requires a nuclear polymerase to interact with the mitochondrial DNA itself. The process of making proteins requires multiple different interactions that are happening in the ribosome and also in the interaction between tRNAs and their tRNA synthetases. This will be sort of the star of the show today, in addition to oxidative phosphorylation. So with all of this potential for interactions between polymorphisms in those two genomes, it's been posited that these two genomes experience a coevolutionary dynamic. And of course, at some level, we know that that must be happening because the mitochondria from us and the mitochondria from an elephant, for example, are not interchangeable. We know that our mitochondrial and nuclear genomes over long evolutionary periods of time are certainly co-evolving with each other. It's more a question of what's happening on a more local scale as populations diverge and as species are acquiring differences as they diverge from each other. To what role is mitochondrial coevolution playing a really important role in shaping that evolutionary dynamic? And one of the frameworks that you usually hear about coevolution between the mitochondrial and nuclear genomes is in the area of compensatory coevolution. And this is because we tend to take a very animal-focused view of mitochondria, and animal mitochondrial DNA have really high amino acid substitution rates, much higher than nuclear genomes. In mammals, it can be 35-fold the substitution rate. And there's several lines of argument that suggest that those mutations that are accumulating in mitochondria are deleterious. Now, the genes in the mitochondria encode some very conserved and important elements of our physiology. And so one model for the maintenance of those functions in the face of these deleterious substitutions is that the nuclear genome can experience selection for compensatory mutations that ameliorate the deleterious effects of those mitochondrial substitutions. And so there could be a lot of coevolution that's occurring simply to keep the mitochondria functioning in their conserved role in oxidative metabolism. So that's a model of compensatory coevolution. The other reason that this has been sort of a rising and increasingly important area of research is that some people have suggested that because of this coevolutionary trajectory, that divergent populations or newly diverging species may be reproductively isolated through these incompatibilities that would arise if you substituted the diverging mitochondria from one lineage into the nuclear background of the other. And so some people have even suggested that these mitonuclear incompatibilities could be important in the process of speciation. So what is our evidence that mitonuclear coevolution actually occurs, other than the fact that we know that our mitochondria work well with our nuclear genomes and better than if we substituted some divergent mammalian mitochondria? Some of it comes from patterns of molecular evolution. So one of my favorite papers, and I think some of the most compelling evidence of this, comes from Osada and Okashi in 2012, where they looked at correlated patterns of molecular evolution between the mitochondrially encoded and the nuclear encoded components of cytochrome C oxidase. And they looked across divergent lineages and primates and really saw patterns that were indicative of this process where you would get a substitution in the mitochondrial genome followed by a compensatory substitution in the nuclear genome. And so they were able to not only see a pattern of correlated molecular evolution, but really one that supported this model of compensatory coevolution. We also have some nice empirical evidence that comes from model species that we can cross between closely related species. So some nice work in yeast that shows that when you do a cross that brings the divergent mitochondria into the divergent nuclear background, you get incompatibilities that can affect things like budding, and this comes from effects on gene regulation. Now, one of the famous systems, and I'm not sure why its picture is not popping up here, but there should be this really adorable marine copepod, Tigriopus. And Tigriopus is probably the system where we understand best the potential role of these mitonuclear incompatibilities in creating reproductive barriers between divergent populations. So in this marine copepod, they have surprisingly divergent mitochondria between populations, even though they're a marine organism. And this is occurring at the level of within species divergence. And you can get hybrids, you can make hybrids between these populations and do it in directional crosses where you know how the mitochondria is moving because of its maternal inheritance, that have really supported that there are multiple incompatibilities between these populations, between their mitochondrial and nuclear genomes. And Ron Burton and his colleagues have been able to attribute that to multiple different functions in the mitochondria, from replication to transcription to the process of oxphos. These aren't just important for our understanding of evolution, but there are many diseases that are associated with variation in the mitochondrial genome in humans. And these mitonuclear interactions, so where you have a SNP in the mitochondrial genome that has differential effects on a trait depending on what nuclear genome it's found in, these can have really important effects on the heritability of mitochondrial diseases. And mitochondrial defects can cause a diversity of human disorders, and they can be relatively frequent, so affecting about 1 in 8,500 individuals. Now, some of these are aging-related, but others are inherited. But many of these diseases are really marked by variable penetrance, where a mother that has a particular mitochondrial variant, while she will pass that variant on to all of her offspring, only some of them may manifest the disease. And some of that may be due to compensatory effects of alleles in the nuclear genome that the offspring inherit differentially from their two parents. Okay, so a while ago now, working with David Rand and Colin Mickeljohn, we decided to do an evolutionary genetic, I'm going to call it an evolutionary genetic screen, where we substituted an increasing amount of mitochondrial divergence into nuclear genomes in Drosophila melanogaster. And so what we really wanted to know was if you move a mitochondria from a divergent population of melanogaster or from increasingly divergent species, do you get worse phenotypic outcomes when you substitute a more divergent mitochondrial DNA? And that would be indicative that these lineages are, in fact, accumulating these types of coevolutionary dynamic between mitochondrial and nuclear genomes. And so this required a lot of brute force fly genetics. So we substituted those divergent mitochondrial DNAs using introgression through hybridization, but then we used all the toolkit of melanogaster genetics to precisely substitute 2 wild-type genomes from Oregon and from Austria. And so when we did that, we were actually quite surprised to find that there was really no indication that a more divergent mitochondria was having a worse fitness outcome when we substituted it into melanogaster. Rather, what we found is that species are segregating polymorphisms in the 2 genomes, that when you brought together particular combinations of those polymorphisms, you would see sometimes rather severe incompatibilities. And so rather than evidence showing that these lineages had diverged and were accumulating fixed mitonuclear incompatibilities, what we saw was that there's the potential that variation within the species can interact with each other to have negative effects on fitness. And it's from that screen that I'm going to show you some mapping data where we were trying to figure out the genetic basis for one of these particular incompatibilities. And it's an incompatibility between a simulin's mitochondria from the simulin's white 501 strain, and it's an incompatibility between a polymorphism that carries and a polymorphism in the Oregon R nuclear background. But first, when we call something an incompatibility, what do we even mean at the phenotypic level? So here I'm just showing you what was the first thing that we really noticed about this genotype was it has a really extended development time. So I'm showing you 3 of those mitochondria here from melanogaster and simulans. And though each of those exists in 2 different nuclear genotypes. So these are the 6 different combinations from pairing those mitochondria with these 2 nuclear genomes. What you'll notice is that the mitochondrial DNA makes no difference to development time if you look within that Austria nuclear background. It also has the same development time if you pair it with either a melanogaster background or if you pair it with a melanogaster mitochondria or a simulans mitochondria. But when you take this SIM-White501 mitochondria and pair it with the Oregon R nuclear genome, you see this really extended development time. Okay, so that's an epistatic interaction or a gene-by-gene interaction, in this case a mitochondrial-nuclear interaction, that has a strong effect on development time. And that same genotypic combination has a 50% decrease in female fecundity. So it impacts the larval development and the adult fitness through reproduction. So that's what we're calling an incompatibility. And those are the types of graphs and phenotypes that I'll be showing you throughout this first part of the talk. So to map the mitochondrial variant, well, that's tricky, but it is helped by the fact that we substituted a lot of different simulans mitochondria, and we substituted some that we expected to be quite similar to each other in their sequence. And SIM21 and SIM-White501, they have a big phenotypic effect, but they actually are quite similar to each other. And together, they share a lot of differences from Oregon mitochondria. And so we were able to say, well, what are the specific variants that differ in SIM-White501 and SM21 that potentially cause this effect? And there's only 5 different substitutions between those 2 mitochondrial DNAs. A big chunk of 3 of them were synonymous. One of them was at a very non-conserved site in the ribosomal RNA. And one of them is this change in the tRNA for tyrosine, and it changes this GC to a GT, which is not a very dramatic change. And in fact, when I did all of this sequencing, I was a little bit disappointed because none of those seemed like particularly strong candidates for having this large of an incompatible effect on these phenotypes. But we went forward and used classic approaches, recombination mapping in the nuclear genome, along with some precise replacement to show that this, in fact, was the nuclear variant that interacted. And what we were able to show is that the polymorphism in the nuclear genome is an amino acid changing polymorphism. It's polymorphic in Drosophila, so it's there at about 13% in at least one population that we've looked at. And it's a change in the tRNA synthetase encoded in the nuclear genome that's responsible for putting the amino acid on this mitochondrial tRNA. So all of a sudden, that mitochondrial polymorphism made a lot of sense in the context of this genetic interaction. So just to summarize that, the 2 variants that don't work well with each other, one of them is a polymorphism in the tRNA, and the nuclear one is a polymorphism in the tRNA synthetase that's made in the cytoplasm, goes into the mitochondria, and physically interacts with this tRNA. So you would expect that this interaction would then compromise translation, protein translation in the mitochondria. And that's exactly what we think it does. If we look at enzyme activities of the different complexes in the mitochondria, we can take advantage of the fact that one of those complexes, complex 2, involves no protein translation in the mitochondria. And for that complex, this genotype that has the incompatibility has normal levels of complex activity. But for all of the other complexes that require protein translation in the mitochondria, which we expect this mitonuclear genotype to be compromised in, we see low activity of those complexes. So this supports that genetic interaction is decreasing mitochondrial protein synthesis, and an outcome of that is that there's low OXPHOS capacity. So working with a number of talented students, we've been using this set of genotypes as a way to understand how these mitonuclear interactions, which are themselves context dependent, right? They're an epistatic interaction between polymorphisms. How is the expression of that incompatibility also dependent on the environment? And we've looked across a number of abiotic environments, as well as differences across life stages and between the sexes. And I'll just give you the take-home message of that, is that the strength of the phenotypic effect of that mitonuclear interaction increases with increasing energy demand. All right, so when energy demand is high, then this mutation that breaks the energy supply, it has a worse effect on the phenotype. And so I'll just give you some examples of what those patterns look like. So this is development time again, and this is the same result I've shown you before. It's just plotted a little bit differently. You can just keep track of the green triangle. That's the mitonuclear incompatible genotype. This number here shows you the extent of the developmental delay of that genotype. And the stars indicate the significance. And so this is where we had done all of our phenotyping and mapping. And thankfully, we did not do any of our phenotyping or mapping down at these lower temperatures, because at these lower temperatures, that developmental delay completely goes away. So this interaction, which looked so strong to us at 25, essentially you wouldn't have even seen it as a mitonuclear incompatibility if you developed these flies at 16 degrees. And of course, for those of you that work with flies, 16 degrees is someplace we keep flies on a regular basis. This is not an extreme temperature. So this means that in the course of a day, sometimes, that mitonuclear effect will be different. And in fact, if we raise them at high temperatures, we can even magnify the extent of that developmental delay. So it's a strongly temperature-dependent interaction. We wanted to try to understand what was happening at the metabolic level, particularly during larval development. So we looked at metabolic rate, and it's plotted here as a function of mass, and the different genotypes are present in different symbols and colors. So at the temperature where there's no developmental delay, we also see that these flies in the green line here have a similar metabolic rate to their nuclear control. So their metabolic rate looks normal. But when you develop them at higher temperatures, where we see that extension of development time, that mitonuclear genotype that has the incompatibility also has a significantly elevated metabolic rate. Okay, so that's interesting. They have compromised mitochondria and oxphos. We actually see that they have this, what looks like compensatory effect, where they respire more, which I think, as Netta showed me, that's sort of related to some of the patterns that she's seen in your lab-evolved strains. So we generally see this pattern of a signature of physiological compensation. So not only do those larvae have elevated metabolic rates, we see that they elevate the level of citrate synthase activity, which is indicative of just oxidative capacity. They also rely more heavily on anaerobic metabolism, so they use lactate fermentation, and that's related to ATP production through glycolysis. And as a consequence of running this kind of higher potentially metabolic rate, on a compromised mitochondria, we see elevated ROS, and those mitochondria have uncoupled membranes. And so this is work that's been done by Omira Montooth, a research scientist in the group, as well as an undergraduate, Jody Thum. So this is essentially an energetically inefficient genotype, I would say. It has a slow development time, but it's running an elevated metabolic rate. So I think what's happening in this genotype is that they're trying to, or they are, maintaining their ATP homeostasis with less efficient mitochondria. What does that mean? That means that that's going to require the utilization of more energy stores. If you think about what an insect larva is doing, it's really doing two things. It's investing in growth, but it's also needing to grow through maintaining its energy stores and accumulating its energy stores. So what we see in this genotype is development is slowed. Basically, the accumulation of mass is slowed, but eventually they get there. So they get to be a normal mass larvae, they enter metamorphosis, and they survive metamorphosis and come out as adults that are essentially wild type in their size. There's several lines of evidence that support that these are less energetically efficient. So this is one of my favorite phenotypes. It's so dramatic. It's how far the larvae will climb out of its rotten fruit before it goes into pupation, which ecologically is probably quite important because that fruit is only going to get worse as it goes through its course of fermentation. Classically, mutants that affect energy metabolism affect this phenotype. And so this height at which the larvae will climb to pupate can be a readout of their kind of potential energetic status. And so you can see that it's quite poor in these incompatible genotypes, and it's highly temperature dependent. So at 16 degrees C, you'll get these larvae crawling up to sort of normal height. One of the things we wanted to know is how much is this environmental effect really the direct effect of temperature, or does it have more to do with something about the energy demand on the organism? And so we wanted to take temperature out of the equation and come up with another environmental regime that would increase energy demand. And so what we decided to do was hold temperature constant, but change the light cycle. So if you extend the light cycle in flies by just giving them a full light cycle, what you'll see is that that increases the rate of development. They'll develop faster. And we wanted to know whether if we could mimic the developmental delay simply by putting them in that long day environment. And it does. So at 16 degrees, these are like the data. These are not exactly the data. It's a different experiment, but these are the data I showed you before where there's no effect of that mito-nuclear incompatibility. But if you give them a full day of light, then you see that incompatibility, that developmental delay at 16 degrees. Also at 22 degrees, it magnifies the effect of that developmental delay, making it even longer than the developmental delay that we were seeing at 25 degrees. So just changing I would say the energetic environment of the organism, the energy demand for rapid growth, mimics the effect of temperature. So I don't think it's a direct effect of temperature. I think it has to do with greater energy demand. So fast development with massive growth like we see in insects places a greater energetic demand on these mitonuclear interactions that are providing the supply. And that's consistent with patterns that we've seen across adult phenotypes as well. So I thought I would just choose one of the adult phenotypes that I just think is a really cool result to show you. We've looked at lots of different adult phenotypes, but one that was worked on by a student in my group, Justin Buchanan. He was interested in energetic effects on immune function. And so he infected these genotypes with a gram-negative bacteria that's a natural pathogen of Drosophila. And in fact, the strains we used were actually taken from Drosophila in the field. And so we infected males and females, and we either did a control or sham infection, or we infected them with the bacteria, and we looked at their survivorship. And what he found was a strong interaction effect where the mitochondrial nuclear genotype survived less well than other genotypes. It looks like there's a stronger effect in females, but there actually wasn't a statistical effect of that. So in the statistical results, the effects of infection on females and the effect of this mitonuclear interaction were additive. But Justin did this really cool thing, and he took all the individuals, females that survived, and he then mated them and asked what was their reproductive success. So across all of our different genotypic controls, if he took infected females and their sisters who had been given a sham infection, and he looked at the number of offspring they could produce over their lifetime, he found that there was no difference. So there was no trade-off between investing in immune function and investing in reproduction in the future. But in this genotype that has compromised energy metabolism, what he found is that those females that had been infected and survived produced fewer offspring than their sisters who had been sham infected. And so what this suggests is that mitonuclear variation, because it can impact the amount of energy resources that are available in the cell, even when these flies are given all the nutrients they need, right? So all of the food nutrients are available to all of these different genotypes. It's this mitonuclear genetic variation that reveals this trade-off between investment in immune function and future investment in reproduction. All right, so that is what I wanted to show you in terms of the phenotypic outcomes and how the environment impacts those phenotypic outcomes of this mitonuclear interaction. So a couple of take-homes from that. I think generally we can say that when energy demand is high, then genetic variation and genetic interactions that compromise the energy supply are more deleterious. And then I showed you that mitonuclear genetic variation may be an important source of variation that can reveal trade-offs between things like immunity and reproduction, even when resources are unlimited. So we often think about these trade-offs as being revealed when the nutrient environment becomes like starvation or limited in some way. But genetic variation can do that same thing at the cellular level. So it can generate a starvation in cellular nutrients, even if the organism has access to all the nutrients that it needs. So now I want to switch to how this research has led to some hypothesis testing at the level of genome coevolution. And part of this came, when we discovered and mapped this genetic variation or this genetic interaction, not much attention had been paid to this important evolutionary dynamic between mitochondrial tRNAs and their tRNA synthetases. So the next part of the talk, I'm going to show you how we've used those protein tRNA interactions to do some tests for whether coevolution is really predominant across animals. And it really hinges on a couple of key things. First is the reality of the fact that in your nuclear genome, you have two sets of tRNA synthetase proteins. These are called the Rs proteins. So you have a set of Rs proteins that are interacting with your cytoplasmic tRNAs, and those fund all of your cytoplasmic protein translation. And then you have a set of mitochondrial Rs proteins encoded in your nuclear genome that then interact with mitochondrial tRNAs in the mitochondria. Now, several groups have shown that mitochondrial DNAs have high substitution rates, and in particular, Mike Lynch has done some nice work to show that tRNAs in animals are accumulating more substitutions in their mitochondrial genome than in the nuclear genome. And some of that work had also indicated that those are likely deleterious substitutions in those mitochondrial tRNAs. So given that pattern, if mitonuclear coevolution is a predominant force shaping genome evolution, then we would predict that given those faster rates of mitochondrial substitution, then the mitochondrially targeted Rs proteins from the nuclear genome should be having a higher evolutionary rate than those cytoplasmically targeted Rs proteins. So essentially thinking about those tRNAs as the driver of coevolution in the protein partners, that dynamic should be going faster in response to these higher substitution rates in mitochondrial tRNAs. And so this was a project I did with Jeff Adrian and Signe White when we were all back at Indiana University. And we went and got a bunch of sequences, all the sequences at the time that we could for these Rs proteins from mammalian nuclear genomes. And we did a phylogenetic estimate of D and DS, so we used omega. And we found that those mitochondrially targeted Rs proteins had significantly higher evolutionary rates of substitution than did the cytoplasmic Rs proteins. Remember, these proteins are all encoded in the nuclear genome. So they have a similar genome that they're encoded in, but they have higher rates of substitution. And that pattern held up across the available bird sequences and the 12 Drosophila genomes that we had available to us at the time. And so this pattern suggested that across these different phylogenetic groups, that substitution rate in the mitochondrial tRNAs was driving a high rate of coevolutionary substitution in the nuclear genome. But of course, many of you who are familiar with patterns of protein evolution know that one of the strongest predictors of evolutionary rate in proteins is their level of expression. And so I had this hunch that cytoplasmic Rs proteins were probably expressed at higher levels than mitochondrial Rs proteins because there's a lot more protein synthesis that happens in the cytoplasm. And so you would predict that that would lead to lower evolutionary rates. And so I felt like we really needed to control for that. So we were able at the time to get expression data for each of those phylogenetic groups. And so if we correct that evolutionary rate for gene expression levels, what we see is that that pattern of elevated evolutionary rate in the mitochondrial Rs proteins goes away if we look at the chicken data or at the bird data or the Drosophila data. But that pattern holds and actually gets stronger when we look in the mammals. Okay, so what that indicates is that those faster rates of mitochondrial R's evolution may be driven by mitonuclear coevolution in mammals, but I think in birds and flies likely reflect selective constraint based on gene expression. All right, what this indicates is that different phylogenetic groups likely experience more or less mitonuclear coevolution. And so why do we think that there's more in mammals? Well, mammals have a really high ratio of mitochondrial to nuclear substitution rates. So really, if this coevolutionary dynamic is driven through this compensatory evolutionary model, you really need the input of those deleterious substitutions to drive significant nuclear compensatory coevolution. And it may just be that in mammals, where we have these high ratios of mitochondrial to nuclear substitution rates, that's where you really see a predominant effect of this coevolution. So here I think it's suggesting that, that some characteristic of organism mutation or repair processes is having an impact on that longer-term molecular coevolution between genomes. So where on the tree of life do we expect to see these significant impact of mitonuclear coevolution? I've shown you some data from phenotypic and empirical work that suggests that probably not in Drosophila. I think they have relatively high purifying selection. They have low substitution rates in their mitochondrial genomes relative to the nuclear genome. But yes, in mammals where you have these high substitution rates. This has sort of motivated us to work on this snail, the New Zealand mud snail, and it's famous for the fact that it segregates for sex. So within lakes in New Zealand, there are repeated formation of asexual lineages of these snails that coexist with sexuals. And so here we can actually ask, how does sexual mode affect mitonuclear coevolution? And that's interesting for a few reasons. One, we know that the asexual lineages accumulate and maintain, they harbor more deleterious polymorphism in their mitochondrial genomes. That's work of Joel Sharbrough down here. And it's possible that the kind of clonal selection you get in these asexual lineages might be relatively strong selection favoring the lineages that link up these favorable combinations of mitochondrial and nuclear variants. And the other reason that they're good to work on from this perspective is that there's significant mitonuclear discordance across the lakes. And so we've been able to sample reciprocal combinations of mitonuclear genotypes. In flies, we can make them, right? But not all organisms out there can you do that type of crossing. And so you need to work with systems that have this mitonuclear discordance that you can essentially sample from. Right, and we have asexual lineages, so we have a bunch of clones. So in some ways, they're also like our mitochondrial genotypes, is we can make a bunch of them and raise them across different environments. And so we're kind of venturing out into this system. I'll just show you one piece of data. So we know that the asexual lineages do harbor deleterious mitochondrial DNA mutations, and more of them than their sexual counterparts. So we thought, well, maybe they would have lower oxygen consumption, but it turns out that they don't. So even across different temperatures, we see that sexuals and asexuals, despite the asexuals having more mitochondrial mutations that look like they're deleterious, they actually can respire at the same rates. And so this lends some support to this idea that clonal selection may be operating to isolate functional mitonuclear genomes in asexuals. And so that's what we're working on in that system. All right, so the bit of unpublished data that I'll show for you here at the end is some work from Abhilash Dewanjawar focused on an interesting pattern that's been observed, which has been called a number of different things. We're calling it compensated disease-associated residues, but these have also been called compensated pathogenic deviations, or CPDs. And I'll show you what that pattern is here in a minute. It's interesting from a mitochondrial perspective because there's a lot of mitochondrial mutations that have been associated with disease. So we're calling these disease-associated residues, and they impact many different proteins in the mitochondria and impact and cause many different types of diseases. But there's this phenomenon, and this is not specific to the mitochondria, that sometimes an amino acid change that's associated with disease in humans is actually the wild type variant in other species. And that's what has been called compensated pathogenic deviations or compensated disease-associated residues. Compensated because that observation leads you to think that probably someplace else in the genome, there's another mutation that's causing that variant not to be pathogenic in that other species. Okay, so let's just look at what we would call a compensated or an uncompensated disease-associated residue. So here we're just showing you some examples from data we've acquired. These are mitochondrial COX-2 proteins. So this would be a conserved site across these species, but we know that there are associated residues, so a mutation here to a Q in humans that's associated with glaucoma. But that's an uncompensated, we would call that just a disease-associated variant in humans, important from a disease perspective, but not that important in terms of thinking about the molecular evolution. But let me contrast that with these two different residues. So here there's a relatively conserved amino acid. We know that in humans there's a disease associated with the mutation to a different amino acid here. And that amino acid is the wild type or sequenced amino acid in the macaque. Or here we know that there's a change in humans, and that change is associated with the disease, and there's actually 3 other species in this group that have that as the wild type variant. And so the inference there is that someplace else in the genome, there's a mutation that causes that mutation not to be pathogenic in those other species. So previous studies that have looked at nuclear proteins have reported that something like 2 to 18% of these human disease-associated residues must be compensated because they're present as the wild type in other species. So Abhilash wanted to know, well, is there something about oxidative phosphorylation that might enhance this dynamic, essentially because there's a lot of space in those complexes for protein amino acids to be touching each other. And so there's a lot of space for this compensatory coevolution to be happening. So this is just an example of one of the things that he's found, and I'm just choosing it as a case study to show you what this dynamic may look like in terms of protein evolution. So this is COX-1 in the mitochondria, and there's a variant in humans at this position where this this mutation to a T is associated with a disease. All right, it's prostate cancer in humans. But that's the wild type allele that we find in the orangutan. So we know that COX-1 interacts quite closely at the structural level with COX-7C. This is a nuclear gene. And so Abhilash looked at those protein alignments, and there's another position in that protein where we see in those 2 species that have that human disease-associated residue, they have a substitution at this amino acid. And so we wanted to look at that in the context of the structure. And so this is really a case example of what you might expect to see with one of these compensated disease-associated residues. So this is the wild type situation in human at those 2 amino acid variants. This is the mutation at that residue 117 in humans, and what that does structurally is it brings those 2 amino acids apart from each other. Right? This is the interface between COX-7C and COX-1. This is part of oxidative phosphorylation. If you look at the wild type situation in Pongo, which has the T variant at 117, but it also has a substitution at this position 55 in COX-7C, you see that that mutation compensates that interaction. It brings it back together. So one of the reasons that people have studied this pattern is that it's indicative of this pattern of epistasis or interactions being important for maintaining the same protein function, even in the presence of substitutions in proteins. All right, so what he really did was not scan individual genes. He went to GenBake, got all the mammalian mitochondrial genomes that he could, did a really big multiple sequence alignment, annotated that with all of the well-known disease-associated residues in mitochondrial oxphos proteins, and then categorize them as being either compensated, meaning that that mutant amino acid is present as the wild type in a non-human species, or it was not present, in which case we just call that a disease-associated residue. In humans, it's not present in any other species. So it turns out there's a lot of mammalian mitogenomes that have been sequenced. So when Abhilash started working on this, there were over 1,000 mammalian mitogenomes, and he was able to categorize 267 disease-associated residues in those OXPHOS proteins. So he's done two analyses. He's basically looked at a phylogenetic analysis, and then he's done a bunch of structural analyses. And I'm just going to show you the data from the phylogenetic analysis today, but he's about ready to submit a paper to bioRxiv, so be watching for that. So of those 267 DARs that are present in those OXPHOS proteins in mammals, 138 of those are compensated. This is just showing you the frequency spectrum. So this is the number of species that have that pathogenic amino acid. So you can see that many of them are rare across the phylogeny, but some of them are quite common. And we can also ask, well, how many times did they arise on the phylogeny? Because we can do this in the phylogenetic context. And some of them arise only once, but some of them have a number of independent origins. Right, so some of these stars are rare and some are common, and some of them have arisen multiple times on the phylogeny. Now, the really striking pattern that we've seen is that while other protein data sets that have been analyzed show about 2 to 18% of those disease-associated residues will be compensated or present in other species, in the mitochondrial OXPHOS proteins, 52% are compensated. So 52% of those disease-associated residues are wild type in another species of mammal. So we thought, well, maybe the reason that we're seeing such an elevated fraction being compensated has nothing to do with them being in OXPHOS or being on the mitochondria. It's simply because we were able to sample 1,000 species. And these other studies, well, some of them don't even report how many they used, so we just called it 50. And others use, far fewer mammalian species. And so what Abhilash did is he just downsampled the data. So these are a bunch of different replicates where he just, out of that 1,000, he selected 700 and reran the analysis. And so you can see that there is an effect of sampling more species, but in these mitochondrial OXPHOS proteins, we never get down below really 30% of them being compensated. So it really seems like these OXPHOS proteins have a higher propensity for this type of compensatory disease-associated residue pattern. For some of these data sets, we could pick out the same mammalian taxa that they had used and only include those in our analysis, and those are the blue dots. So we can see that even when we restrict ourselves to the exact same mammals in the analysis, we still see this elevated rate of compensated DARs in OXPHOS proteins. Now we could ask, is that something specific to OXPHOS, which was kind of our working hypothesis in the beginning, because OXPHOS has all of these potentials for protein amino acid residues to interact with each other. And so, but it's actually not that, because we can separate those into how many are mitochondrially encoded, and then he was able to go into the database and get a lot of nuclear encoded OXPHOS proteins, and you can see there's a big difference. So it goes from 52 to 13% being compensated. And in fact, there are 57 species where we have both the mitochondrial and the nuclear data. And in those, we can see that 33% of those mitochondrial DARs are compensated, whereas only 6% of the nuclear DNAs are compensated. So this is like, these are all proteins that are operating in the same OXPHOS system environment, but it's something about being encoded on the mitochondrial DNA. And if I was going to answer why, it would be a lot of speculation. So I think that's something that I'm looking to see if anybody there has some good ideas. And so with that, I'll just give you my take-homes, and we can talk more about that finding after the talk. So energy demand may be a general explanation for these context-dependent genetic effects that we see across life stages, across sexes, and across environments. those mitonuclear interactions, or in general, variation in energy metabolism, may be really important in thinking about how that variation generates context-dependent life history trade-offs. I also showed you from molecular patterns that mitonuclear molecular coevolution may have its largest impact on species that have elevated mitochondrial substitution rates. And I've shown you that the mitochondrial genome itself may be a unique evolutionary environment for mutations to experience selection. And with that, I tried to thank people along the way for their contributions to my research, to the research I showed you today. But there's a lot of people who contribute to our thinking, both in the lab and our collaborators. And of course, thank you to our funding resources. And I'll just leave you with an announcement slides while I take any questions that you might have.